My name is Nicole Angelique Castro with my thesis partner Ahyessa Castillo. It is both an honor and pleasure to stand before you to present the culmination of our journey. Without further ado, let's delve into the world of biogas and explore the findings and insights that have emerged from this thesis presentation.
Let's start with an introduction that sets the context of our discussion. In recent decades, waste generation has continuously surged worldwide, and there is no evidence of it slowing down. Globally, municipal solid waste volume is likely to have grown by 70% to 3.4 billion metric tons by year 2050. This is related to a variety of causes, including population increase, urbanization, economic growth, and consumer purchasing behavior. Waste minimization is a solution, though waste cannot be fully avoided. The second best alternative would be to recover resources and energy from this, as well as to remanufacture and recycle garbage into usable goods. Household and agricultural waste accounts for most of the municipal solid waste in several nations. When food waste is collected individually, this form of garbage can be used as a source of energy like biogas or fertilizer. This can help achieve all 17 of the United Nations Sustainable Development Goals.
Biogas is a naturally produced gas that is generated by anaerobic bacteria when organic waste is broken down and utilized for energy generation. Anaerobic digestion is a method of breaking down organic matter, such as food waste, in the absence of oxygen. Today, the non-toxic iron-based materials, such as iron, iron oxide, and salt additives, were considered as an efficient not only because of the cost of these materials, but also it is a great additive to enhance the production of methane and sulfate management from the anaerobic digestion systems. Numerous investigations have been conducted under varying operating conditions to improve the production of biogas by the enhancement of microbial activity through the application of diverse chemical and biological additives. The performance of biogas plant could be considerably increased by adding these additives.
Our study has the potential to contribute valuable insights to anaerobic digestion because for anaerobic digestion of pineapple wastes, the optimal concentrations and mechanisms of ferric chloride actions have not yet been identified. In addition to that, most of the studies covers the digestion of pineapple waste, where there were no studies that covers pineapple waste as a substrate used in anaerobic digestion with the effect of addition of ferric chloride. Lastly, there has not been a study of identifying the appropriate kinetic model for the anaerobic digestion of pineapple wastes.
In this study, the effect of adding ferric chloride in the pretreatment substrate will be evaluated based on the biogas yield. In addition, the comparison between the monodigestion of the pineapple waste that contains only the pulp and the monodigestion of the whole fruit of the pineapple waste as a substrate will also be determined. In order to identify the best fit model for kinetics research, a comparison analysis of the models was conducted.
Once the optimal parameters for setting up the anaerobic digestion of pineapple waste have been identified, the efficiency of biogas will be maximized using its components, which are core, peel, and pulp. Also, the findings will be sufficient to select appropriate kinetic model for forecasting methane generation during anaerobic digestion. It will also serve as a resource for academics studying theory.
Despite the extent of the researchers' interest, the research is only limited and only concerned with determining the effect of the addition of ferric chloride on the anaerobic digestion of pineapple waste only, and it will be done in 4 batches and kinetics. Additionally, the value of biogas for sanitation and the proper volume of ferric chloride will not be taken into consideration. Additionally, because there are still many pretreatment methods that might be applied.
In order to conduct the experiment, the methodology consists of 7 parts, starting from sample collection and preparation of inoculum and substrate up until to the kinetic study. For this step, pineapple waste is obtained from Dole Pineapple Plantation in Palomaloc City, South Cotabato, 3 days before the anaerobic digestion takes place. To ensure that the pineapple is still fresh, the pineapple core, pulp, and peel are the parts of the pineapple that were crushed and blended for this process. The bacterial inoculum, which is composed of cow manure, and distiller wastewater has a mixing ratio of 50-50 and is stored in an ambient temperature for 7 days. The requisite nutrients were obtained from Mapua University Chemistry Laboratory located in Intramuros, Manila, and type 3 water is used to dilute the micronutrients into substrates needed.
This images shows the preparation of the sample collection, which is the pineapple waste. A series of 4 laboratory-scale reactors consisting of HDPE bottles with a volume of 1 liter were prepared, which demonstrates in Figure 1. A 1 liter oxygen gas bag is used to link to every media bottle via a rubber tube for biogas collection. The digester is then sealed off from outside air and depleted of oxygen so that the archaea bacteria may properly carry out the methanogenesis process and generate methane and carbon dioxide.
Mixed substrates and inoculum are stored in 1 liter HDPE bottles with a mixing ratio of 80-20. The first run is composed of mixed pineapple pulp and pineapple peel that serves as a control for the following batch, C1. The second run is composed of the pulp and the pineapple waste, R1. Third is composed of mixed pineapple pulp and pineapple peel with a concentration of 150 milligrams per liter of ferric chloride, R2. Fourth is composed of mixed pineapple pulp and pineapple peel with a high concentration of 250 milligrams per liter ferric chloride, R3.
The substrate that entered the digester is analyzed for pH, total solids, and volatile solids using a pH meter. The starting and terminal pH values were measured and adjusted using calcium hydroxide. According to the literature, a pH of 6.5 to 7 implies that the digester is working correctly. Subsequently, micronutrients were added to the digester, which was comprised of cobalt, copper, iron, magnesium, manganese, and zinc trace elements, and their corresponding amount is shown in Table 2.
The TS and VS content of the solid waste affects the effectiveness of the anaerobic digestion, particularly in the production of biogas and methane. The TS and VS were analyzed in the laboratory using the EPA method 1684 before and after the digestion process takes place. TS These are the pictures when TS and VS analysis were conducted.
The digester is being connected to the setup to save the gas produced after nitrogen purging. The media bottles were shaken at a constant temperature of 45 degrees Celsius on a hot plate. The bottles were left out at ambient temperature for 15 days to digest. The volume of biogas accumulated was measured every day to measure the biogas generation of the whole equipment.
For the analysis of pH, it is carried out before and after the anaerobic digestion process takes place. At the beginning of this process, the pH value of each reactor is adjusted to 6 to 7.5. The ideal pH of 6.5 to 7 is a favorable condition given that it stimulates bacterial growth and it yields more methane other than other pH levels. The findings indicate that the highest amount of biogas produced is produced at an initial pH that is approximately equal to 7. The largest volumes of biogas are produced by reactors 2 and 3, whose initial pH values are close to neutral, which consists of 2103 ml and 1488 ml respectively. Reactor 2, on the other hand, has an established alkaline pH and generates the least amount of biogas, which has an 550 ml. Different pH values may negatively affect metabolism by changing the chemical balance of enzymatic process or by outright killing the enzymes, which is the reason why most microbes thrive best in the environments with neutral pH. As also observed, the pH of all the batches declined from close to its optimal pH to acidic pH, which ranges from 3.3 to 3.52. This could be due to the amounts of volatile acid production that tends to lower the pH of the digester liquid.
The reduction of total solids was also investigated. Total solids decrease indicated that the feedstock solid material has undergone anaerobic digestion and has been converted into biogas. In the figure, it shows that the addition of ferric chloride to reactors 4 and 3 resulted in a discernible total solids and volatile solids removal. The highest biogas yield had a percentage of volatile solid reduction of 93.69%. In the reactor 1, which contains only the pineapple pulp as a substrate, yields the lowest total solid reduction, meaning that high fiber content is highly resistant and is resistant to microbial breakdown, especially lignin. This may impede the organic matter's breakdown during anaerobic digestion or composting, which would reduce the volatile solid reduction. Ferric chloride demonstrated excellent removal of suspended solids from initial total solids values of 93% and 88% for the addition of 250 ml and 150 ml of ferric chloride respectively. This is supported by the study in which at a dosage of 93 milligrams per liter of ferric chloride similarly showed a good removal of suspended particles from initial total solids values of roughly 100 to 110 milligrams per liter. In addition, it is stated that ferric chloride is more efficient than alum for increased total solids removal, according to a study on enhanced removal of heavy metals in primary treatment. The setup represented in Figure 4 in the previous slides was utilized for all 4 runs of the kinetic study. The volume of water displaced over the 20-day period are fitted to the sigmoidal growth functions of logistic functions, modified Goppert's model, and the modified Richards model. Based on the results, MRM is found to be the best fit for biogas production from the anaerobic digestion of pineapple waste under specific conditions. Researchers found that the MGM models predict that the biogas potential that equates to be the rate of bacterial growth in the digester. Furthermore, the findings of the batches containing the ferric chloride, which are the reactors 3 and reactors 4, have the lowest value of lag phase, indicating that among the others, their peak performance of biogas production was obtained in less than 3 days.
For the cumulative biogas measurement, ferric chloride boosted biogas production at an appropriate dosage of 250 milligrams per liter. Iron is one of the most required metal elements for methanogens, which makes the supplementation of ferric chloride in the anaerobic digestion process makes a prominent performance in the methane production, sludge reduction, and sulfur control. Reactor 4 yielded the most overall biogas with a composition of 250 milligrams per liter of ferric chloride and pineapple waste. Biogas is produced in reactor 4 is in 2,103 milliliters. Additionally, reactor 3 produced 1,488 milliliters of biogas with a composition of 150 milligrams per liter of ferric chloride with pineapple waste, resulting in the next largest biogas production, where over 814 milliliters of biogas are produced by reactor 1. Biogas production consists of primary pineapple juice. The control, which is the pineapple waste or the whole fruit generates the least amount of biogas, which is 550 ml. The improved biogas yields in reactors 3 and 4 could be attributed to the presence of appropriate levels of ferric chloride relative to the groups that did not include ferric chloride. Reactor 4 has the highest percentage increase to the control by 282.36%, while reactor 3 has percentage increase to the control by 170.54%. The phenomenon was confirmed in the previous study that the maximal cumulative methane generation at 200 milligrams per liter of ferric chloride was 28.9% more than the control. Iron functions as a catalyst to increase the anaerobic co-digestion process ability to produce biogas from organic waste, which is why this phenomenon occurred. The function of trace metal ferric chloride in the anaerobic digestion process demonstrates that it serves as a micronutrient that for microorganisms in anaerobic digestion systems, promoting their growth and enhancing the production of biogas and removal of pollutants. For the reaction of ferric chloride and ammonia or ammonium nitrate, an aqueous solution of ammonium chloride and iron nitrate is created when iron chloride reacts with ammonium nitrate. Anaerobic digestion breaks down organic wastes, releasing phosphorus and nitrogen molecules into the surrounding environment. Therefore, high amounts of ammonium nitrogen found in the supernatant of the anaerobic digestion process prevent the growth of microbes. When the addition of ferric chloride was added, the concentration of ammonium nitrogen increased the least. Additionally, volatile fatty acid buildup brought on ammonium might further impair the stability and effectiveness of anaerobic digestion. In addition, stated by Evgenovi, that the addition to stimulating methanogens, which produce methane for anaerobic digestion, the addition of ferric chloride to anaerobic digestion slurry improved the development of microorganisms at the chosen concentration, which increased the biogas generation. Subsequently, C1 or control 1 has the lowest biogas yield of 550 milliliters, following the reactor 1 with a biogas yield of 840 milliliters. This shows a discrepancy between the experimental result reported in the study of NAMS3, in which pulp and peel have a higher biogas production than that of only pineapple pulp, accumulated a gas volume of 178 milliliters per gram and 96 milliliters per gram respectively. This can be inferred that the development of bacteria responsible for producing biogas was dependent on certain nutrients obtained from both the pulp and the peel. With regard to the result, the pineapple pulp and the pineapple pulp has the potential to be employed as a biogas producing substrate. It is unable to, however, yield more biogas than when ferric chloride is added. Concluding the study examined the effects of the addition of ferric chloride in the anaerobic digestion of pineapple waste. The results indicated that the addition of ferric chloride enhanced the efficiency and stability of the anaerobic digestion of pineapple waste. Reactor with the most biogas of 2,103 milliliters and 1,488 milliliters produced where the reactor where ferric chloride is added with an amount of 250 milligrams per liter and 150 milligrams per liter of ferric chloride, respectively. The substantial decrease in total solids of the reactors with ferric chloride indicates the improved biodegradability of the solid matter, which allows the microbes to access and break down the organic matter more efficiently, leading to an increase in biogas production. It also shows that a high volatile solids reduction from the reactors with ferric chloride helps in enhancing the production of biogas due to the increase in degradation of organic matter by stimulating the metagonic activity. Moreover, the best fit model for the study in terms of biogas potential is said to be modified Gumpert's logistic model and Richard's model. Overall,
Overall, the results experimentally demonstrate that the dosing of ferric chloride is a strategy to improve and optimize anaerobic digestion process, maximize biogas production, and ensure the environmental sustainability. To safeguard against any possible detrimental effects on the anaerobic digestion process, ferric chloride dosage needs to be closely monitored and controlled. It is advised to regularly analyze and modify ferric chloride levels to guarantee the overall stability and efficiency of the biogas production process, because improper application or overuse of ferric chloride may result in harmful effects such as the introduction of harmful chemicals or disruption of microbial activity. When thinking about using additions like ferric chloride in biogas systems, it is advisable to speak with professionals in wastewater treatment and biogas technology.